Okay, today in medical students arena, we're going to have introduction to microbiology and classification of bacteria. Actually, microbiology is a discipline of medicine taken by the MBChB student, the MBBS, and the clinical medicine and surgery students. And at most, even the nurses they do the same for the first years of their learning. So today we're going to have introduction to microbiology, and the most key thing here today. So right away, we'll start by the milestones in microbiology. How far microbiology has evolved ever since the establishment of the discipline. So beginning from the ancient moment, actually, organism people used to be in a primitive life. I mean, exaggeratively, that religion was controlling everything, and even a deceased state was called upon to be a curse from either gods. Yeah. So treating somebody, maybe giving out an act of management or medical in a way will be a curse to you too, because you are correcting what gods had cursed. So people could leave you to die because of that exaggerated notion. So ancient time, during the medieval times, microorganisms were unknown, and their effects also were attributed to divine judgment like magic or sorcery. But around 1674, when our education came in, evolution was in the spearhead. Things were no longer in black and white, but now some colorful, like the monarchical moment during the Aristotle age, when the diagrammatic representation of our anatomy, even digestive system, was just a sketch from imagination and some notion like idea, but it wasn't true, because even opening a human being's body, like anatomizing or dissecting, was evil in a way. But then in 1674, Anthony van Leeuwenhoek observers microorganism came on animalcules. They called them microorganism animalcules, and report them to the British Royal Society. Then later on in 1798, the GINA uses the first vaccine and begin a process that will lead to the eradication of smallpox in the 1970s, because our smallpox already manifested during that time as epidemic. In 17th century to 19th century, the theory of spontaneous generation of centuries was slowly disproved. And what is this theory of spontaneous generation? This was a stated theory that everything just happened to be in a spontaneous way from nowhere, just in existence at once. So in 17th century, 19th century, it was already started disapproved slowly, a process which was finally completed by Pasteur. I am sure you know the Louis Pasteur and the Tyndall. So in 1860s, Pasteur furthers the germ theory of disease by his work on silkworms and developed pasteurization. Up to now, it's being used even in KCC. Like last week, I was in KCC and I could drink a lot of milk there, and I saw all the process. It was nice. So pasteurization is something done there. 1870s, Lister uses antisepsis. Lister uses antiseptics to control surgical infections, because before then people could undergo surgical operations and they end up dying even after the problem had been corrected surgically. They still die because people are like, what is happening? Why are these people still dying? People came up with very serious conditions, serious infections after the surgeries, and nobody could attribute to any conceptualized based knowledge on what could be going on. But then Lister realized there was some sepsis thing, infection from the unsterilized item, the instrument that they were using. And so they came up with an idea of antisepsis, so controlling, making the environment so sterile so that the infection cannot habitate over in the wound and the sutures which are formed after incisions. In 1876, the Koch, Robert Koch, demonstrated that anthrax is caused by Bacillus anthracis. And then in 1881, he suggests agar to solidify growth barrier for bacteria. So Hayes suggests that agar can solidify growth media for bacteria, and agar is currently like one of the special media which is used in culturing bacteria when in the lab for investigation and sensitivity purposes. So we culture them in the agar. That is Lina in 1881. Then in 1880s to 1900, the golden age of microbiology. So here, many pathogens first were identified. And in 1940s, development of antibiotics begins. In 1940s, still present up to now, widespread use of immunization leads to huge reduction in illness and death caused by many previously common infections, e.g. measles, diphtheria. And in 1980s, development of molecular techniques for diagnosis and engineering begins. I'm very sure, and I always believe that God was so handsome enough Now the creator was so handsome enough to know when to bring me to earth. I don't know. I don't believe in reincarnation, but I'm sure that this is my first life. I will really hate it when I could be in those soul life form. Like when the bacteria, people never knew why they are dying, you know. Forms of life. Let's start with the forms of life. So categorically there are more. We have the we have the Eureka. We have different types of this shit, yeah. But we are going to base our discussion on the eukaryotes and prokaryotes. The eukaryotes and the prokaryotes only. The chair, we're going to leave them, right? All those ones, the gram negative shit, we'll meet them later on, but here we're going to have a basement on the eukaryotes and the prokaryotes only. So on this one, I needed to get it clear that there are higher forms of organisms, and that is eukaryotes, and prokaryotes are simple forms of organism. And then eukaryotes, they have complex cellular organization. That is like your body is eukaryote. So in large scale, I just want you to know that before I say eukaryotes are animals or organisms, so they have the nuclear membrane enveloping their nucleus. So their nucleus contents are not widely spread or floating over in the plasma and cytosol. The same or different from the prokaryote, which they don't have the nuclear membrane. Therefore, their DNA and the hereditary materials are everywhere in the cytoplasm. So in short, eukaryotes are more developed than the prokaryotes. So when you're giving difference of these two life forms, that in eukaryotes they are high forms of organisms, prokaryotes they are simple form of organism. Eukaryotes they have complex cellular organization, prokaryotes they have simple cellular organization. Their nucleus in eukaryotes have membrane bound, and they have no nuclear membrane, that is in prokaryotes. The replication in eukaryotes process is complex due to the nuclear membrane. They have no histones, and I'm sure you know the histones in DNA replications. The ones normally associated with the cell membrane. Then their size in eukaryotes 5 to 10 meters, that is size in 0.5 and 200 in prokaryotes. Ribosomes types in eukaryotes are 80S and 70S, while those ones in prokaryotes are only 70S. Examples of eukaryotes are animals, fungi, algae, and protozoa, while the prokaryotes are the viruses, the bacteria, and the form rickettsia, mycoplasma, and the chlamydia, aclea, all those ones. So let's talk about the Koch postulates. Koch came in. This Koch was a great guy, yeah. So he came in and established if an organism is the cause of a disease, the rules which could be used to confirm that organism is really the cause of the given disease. So that was what was pronounced or named to be Koch postulates. So Koch postulates were just independent ideas that could rule in or out whether this organism, because after the era of microbiology initiated, most organisms were identified, and therefore they could have some similarity in terms of their pathogenesis, and thus brought in Koch postulate to narrow diagnosis or maybe be rest assured of what is really causing the disease instead of guessing. So first, number one, Koch postulate was that it was like an organism must be constantly present in the lesion. So that is lesion, tuseme kama kidonda, ukitafuta organism lazima uipate hapo. B, the organism must be covered from the animal and grown in pure culture. So it can be recovered from the animal and grown in pure culture, to mean that the organism can be taken into the lab and then it is grown there, and it has to grow just the same way that the way it grown the human or that organism. So organism again from the pure culture should produce the disease when inoculated into healthy susceptible animals. So this organism that was removed from the animal and grew in pure cultures can be taken back to the animals and produce the same disease as it produced before. Then final, the organism should be recovered from the experimental infected animal in pure cultures, right? So the organism can this animal now which has been oculated or inoculated with the organism should again be harvested. The organism can be harvested from it again. So Koch later went on to prove the etiological agents of cholera, plague, and tuberculosis by the same postulate. So that is that. So let's go direct to medical microbiology, and here we have to see the study of subject entails the following. 1. Etiology. And what is etiology? Where the organism is found and other characteristics that result to causation of the disease. Pathogenesis. This is the mechanism of how it attacks the tissue or the disease mechanism. Then the clinical presentations are either signs and symptoms, and you need to know signs are subjective. Signs are objective, to mean you can see them. Symptoms are subjective, to mean that they're feelings only to the patient. Laboratory diagnosis comes in later on, which that is microbiology. So after talking about etiology, pathogenesis, clinical presentations, basic of the signs and symptoms of those diseases, we check on the laboratory diagnosis. How now we can evaluate this organism from the lab and indicate or give out indications, and also use this diagnosis to evaluate whether it is malaria, it is amebiosis, it is gardiosis, it is whatever. So that is laboratory diagnosis. Then treatment of infection. After now evaluating and knowing the disease, it is cholera. What are the regimens which are supposed to be used in terms of pharmacological treatment and non-pharmacological treatment? Epidemiology as well. Epidemiology based on distribution and frequency of distribution of the disease from based on male, gender, places, environmental factor, geographical distribution, etc. Then control and prevention comes in at the last thing. That is entirety of what is microbiology entails, right? So roles of microbiology in the community is just microbiology is mainly concerned with the control and prevention of disease. It also deals with the factors influencing infection in the community, which include 1. Overcrowding, 2. Nutrition, 3. Contamination of food and drink, atmospheric pollution, civil unrest, post-election violence and displacement, IDP, that is internal displacement people, and poverty. Those are the factors influencing infection in the community. Overcrowding, mkiwa wengi, the tendency of passing or transmitting infection from one person to another or one household to another is high when there is overcrowding. And again also, clean hygiene is not there in overcrowded places because of lack of adequate space for disposal of the wastes. Nutrition as well. Undernutrition result to other nutrition deficiency diseases like kwashiorkor, marasmus, etc. Overnutrition also comes in with the other lifestyle diseases like diabetes, so you should take care of those. Then contamination of foods and drinks, yeah, that's another thing. The atmospheric pollution, people who rest or live or habitat areas which have got several dam sites, several industry which emit unwanted like sulfur dioxide into the atmosphere. Result to that. Civil unrest, post-election violence, yeah, displacement and all that. Branches of microbiology is the next thing. There are branches more than enough, but we're going to check on the 6. Bacteriology, virology, mycology, protozoology, helminthology, and entomology. I've said we are going to have bacteriology, virology, mycology, protozoology, helminthology, and entomology. Start with the bacteriology, scientific study of bacteria and the disease caused by bacteria. Then virology, scientific study of viruses and the disease caused by the viruses. Mycology, the study of fungi. Protozoology, the study of protozoa. Helminthology, the study of worms. Then entomology, the study of arthropods like insects. So ecology of microorganisms, that is environmental attribute of how they relate and relationship between the environment. That is ecology of microorganisms is how they relate with one another in the ecosystem. So free living includes saprophytes found in the soil, water, sewage, plants, and similar environment that are free living microorganisms. Commensals found in animals, including mans, like ruminant animals. They always have this kind of bacteria. Then pathogens, that they're the one that causes disease versus non-pathogens that do not causes, can't be useful or they are not causing a disease. So classification of bacteria. The classification best considers the following characters: 1. Anatomical, physiological, and serological, ecological. So it's based on anatomical factors, physiological factors, serological factors, and ecological factors. Another important classification consideration is pure cultures. Yeah, the essential pure culture that are essential are clones, strain, family, genes, species. Those are helping us in classification. So we'll see, for instance, bacterial identification classification. Now we're going to have colony, appearance, shape, the ones which are best classified based on the shape. We can have cocci, yeah, the bacilli, spiral, so bacilli, rod-like, cocci, comma-like, spiral, Then arrangement, they can also be classified according to the arrangement, like single, pairs, yeah. You see diplococci, streptococci, single, pairs, whatever, chains and clusters, Higher size 1 to 10 micrometer. Then the most things, and we can also classify them is in depending on the stain that they form. Like when the Christian gram brought in the gram stain, it helps us also in classifying bacteria. So we have gram positive and gram negative. The ones which could turn positive with the gram stain, we go call them gram positive, and the gram negative also, depending on the color, the crystal violet and the purple color. Aerobic versus anaerobic. Aerobic are the ones which now can stay without, with oxygen and not without oxygen. Then anaerobic, they can exist without oxygen. Then physical and structural characteristics also can come into play. Biochemical characteristic, DNA analysis, etc. Morphologic classification. Morphology includes size, shape, and arrangement. Cocci, diplococci, you find them there, right? So based on their shape, arrangement, here is what we're gonna have. The coccus, single one. Diplococci, there are two. Diplococci encapsulated, there are two, but they have capsule. Staphylococci, they are clustered. Streptococci, they are chain-like. Cassina, they are like 4 pairs of. Then you have tetra, they are only 4. Then bacilli, we have Coccobacillus, Bacillus itself, Diplobacillus, Palsides, Streptobacillus, and if you use the YouTube, you can see whatever I'm saying, and you can be able to see them in details. So that is that. Let me try to enlarge this. Okay. So that is that. About size, you can come to check on later. The bacteria are approximately 0 to 5, 1 to 0 micrometer in diameter. That is cocci, 0 to 1 to 3 micrometer with 0 to 5, depending. So can be different variation based on types of bacteria like cocci and bacilli. Yeah. The important consequence of small size eye surface area volume ratio. The large surface to volume ratio through which nutrients enter the waste leave the cells compared to the small volume of the cell substance to be nourished accounts for the usual high growth rate and metabolism of bacteria. Also, due to high surface area to volume ratio, the mass of cell substance to be nourished is closely to the surface, requiring no circulatory mechanism to distribute the nutrient. Therefore, you can tell that the bacteria with high surface area to volume ratio are so little than this one. That one. Then another thing is shape. Shape is governed by the rigid cell wall. Typical bacterial cells are cocci, that is singular. Cocci is plural, singular is coccus. Spherical cells, that is e.g. Staphylococci, Streptococci, Neisseria species. Then on bacilli, we have singular bacilli, tetraodes, Clostridia, Pseudomonas, and members of Enterobacteriaceae. Pleomorphic. Then based on the arrangements, bacterial cells are usually arranged in a manner characterized of their particular species. However, not all cells of species may be arranged in the same manner. The predominant arrangement is the important feature. So like for the cocci, appears in several characteristics arrangements, and depending on the plane of the cellular division and whether the daughter cell stays together following divisions, common arrangements encountered include: we can have diplococci divide in one plane, streptococci divide in one plane with most daughter cells staying together, tetrads or tetracocci divide in 2 planes, then staphylococci divide in 3 planes in regular patterns, Sarcina divide in 3 planes in irregular patterns. So bacteria can be characterized as follows: spheres, these are termed as cocci and can be formed of clusters, chains, or pairs and tetra. What you are able to see is what I'm talking about. So I think we can proceed from this area later. That will be just an introduction aspect of the bacteria, though it's not over. It's not actually over. The images there are now remaining of images of those cluster of cocci, staphylococci, how they look like when you view them using electron microscope or light microscope. So we have other categories that are rods. They are termed as bacilli, and they take shape of a cylinder, fusiform like. So most bacilli, this thing loads slowly. So most bacilli occur singly or in pairs like diplobacilli. Some species form chains like streptobacilli. Bacillus subtilis or Bacillus subtilis is an example. Some species have cells lined side by side like matchsticks. The example are palisade arrangement, that is Corynebacteria, like for example diphtheria, Corynebacteria diphtheriae. Some species form long bronchod multinucleated filaments like hyphae or mycelia, and example is Streptomyces species, and some species form curved like vibroid cells like Vibrocolerae. We have Parahemolyticus, some are helical, spirochetes, flexible, yeah, Treponema species or rigid helicals, Parallema species, and other shapes or ranges also curved. So whatever you are viewing are the images of the rod-shaped bacilli that you can see them, the Fusobacterium. That is that. The loading is slow, but we are good. So I think for that up to here, we are categorically going well. Whatever you're able to see there, the image of vibro curved into comma shape. These are palisade arrangement, the way I talk about. Spiral curved longer than a comma and take a spiral shape. Filament with branching. And it's done. So have a nice time. Don't forget to subscribe.